Okay, what we'll do then is I will lecture till about 1:15, 1:18, or a convenient point in there. I want to get through this lecture, so I want to start with my guys on Tuesday next week. And then we'll stop. We'll look like a pretty well before we do stuff, and we can go now, and we'll get into anything over. All right? So the last time we met, I believe we were talking about heat at the means of sterilization in this. We talked about an autoclave. You recognize the fact that an autoclave is nothing more than a very fancy, very expensive pressure cooker. It allows us to attain temperatures much higher than 100 degrees centigrade because we have pressure associated with it. That pressure is one additional atmosphere. Okay, now then, we talked about the means of assessing sterilization. And you've all seen in the laboratory, you've seen oil-wrapped containers with this type of tape on it. It's called an autoclave tape. This particular vessel, can't tell whether it's obviously a beaker of some sort, but it has been sterilized. And I can tell that it is sterile because of the hash marks on that particular piece of tape. Now, again, this is only sterile until somebody takes the cap off. So then at that point, right, then we don't know. We like to think that it's totally accepted, but it might not be. So if we took it off and put our hand in there and put it back on there, then clearly it's not going to be sterile. But we know that that's an indication of sterility. Another one that we frequently use today is called, is a modification of what we call a spore strip. Now, back in the days before we had plastics, one way to assessing sterilization was to take an organism that produces endospores. Now, we know that endospores are very heat resistant. We've already established that in the lecture, in the previous lecture. But we don't just take any endospore producing organism, such as Bacillus. We want a thermophile. We want an organism that is very heat resistant to begin with, grows best at high temperatures. And we have every right to believe that the endospore that that particular cell would produce would be very heat resistant. And that is indeed the case. The one that we use is called Bacillus stereothermophilus. So we would take this organism and dry it down into a little wet powder. And then you would impregnate it onto a piece of what is essentially masking tape. And we place that masking tape, just like we would place this tape over a piece of aluminum or a piece of foil. Put it on the materials that we want to determine or assess sterilization, and sterilize it autoclave. So no hot air running. But when it came out, we would then take that piece of tape that has those endospores on it and put it into a medium in which those endospores, if they're still alive, if they're still viable, could germinate and go into vegetative growth, which after 24 hours incubation, we would see turbidity in the tube. We would know that that particular material was not sterile because the endospores had not been inactivated. Now that took a lot of work, and as technology progressed, we came up with this. And I actually had one of these, but I didn't bring up my little bag of toys. But you can see the size here based upon its relationship with Sharpie. But sitting right underneath this little capsule, this little capsule, this plastic cap, is a little capsule, a gelatin capsule that is filled with endospores from Stereothermophilus, Bacillus stereothermophilus. So you place these into the autoclave. No tape. Just put them in the autoclave. Various portions. One in the back, one in the middle, one in the front. And then when the autoclave run its cycle, you'll take them out and you turn it upside down so that the white cap is down, and you wrap it hard on the hard surface. That breaks that gelatin capsule, releasing the endospores into that medium. Now, I don't know how clearly you can see that, but that's an orange-colored medium. You put that into the incubator. If in 24 hours you come in and that ink, after incubation, that medium has turned yellow because of the acidity produced by the organism. As it grew, you knew that the organism was viable, therefore the material in which you had placed that particular capsule was not sterile. This is one way that many hospitals have to verify on site the efficiency of that autoclave. I believe I also mentioned not that this material so that they know that they've been subjected to qualification. Okay, let's move into radiation. We're not going to spend a lot of time with radiation. We could talk for 4 or 5 lectures on radiation if we wanted to, and that's not our purpose here. But we can use electromagnetic radiation as a means of controlling microorganisms. Now, I think you know what electromagnetic radiation is. Many of you have some high school physics, at least I'm certainly going to talk about it then. But we are constantly being bombarded by all sorts of electromagnetic rays. Radiations of different wavelengths, of different intensities of energy. If we brought a transistor radio in here and turned it on, you could dial to different stations because that radio is receiving different electromagnetic radiation of different wavelengths. Each radio station has a different wavelength through which it is broadcasting. And we, as a receiver, we can pick it up. All you have to do is look around the room and see the different colored materials that everybody's wearing. Visible light is a form of electromagnetic radiation, and different radiations in the visible electromagnetic spectrum give us different colors. And then we go farther and farther down. Now, we can get into some of the rays which are very, very dangerous because of their short wavelength, high energy. And that gets us into this one right here. You do not have this. It's just an explanation thing. I think everybody, if you really need it, I'll give it to you, but I don't think it's necessary. Here we see 6 different types of electromagnetic radiations. They all differ in the wavelength that each exhibits. Longer wavelength. A wavelength is measured from crest to crest, or if you want it, from trough to trough. From crest to crest is the wavelength. Now, this shows us a very long wavelength. This might be a radio wave, in which it might be measured in terms of thousands of meters. It is that long. This is shorter wavelength, shorter yet. We get down to here, we see very short wavelengths. There is a relationship between the wavelength and the amount of energy that that particular form of electromagnetic radiation possesses. High wavelength, long wavelength, large wavelength, low energy. Shorter wavelengths have much higher energy. So we talk about low wavelength radiation. Typically we're talking about ionizing radiation. Ionizing radiation is down here what we would call gamma rays and X-rays. It's so called because it is so energetic. Materials that are exposed to it, it will bounce electrons out. It produces ions. A little less energy up here. You see, we're going by wavelength. That's 10 to the minus 5 nanometers. That's 10 to the minus 9 meters. Here we have ultraviolet light, a little bit longer wavelength, but still has some energy in it. UV light, what we call non-ionizing radiation. Ultraviolet light is non-ionizing radiation. Still can be used as a means of controlling microorganisms. Principally because the wavelengths that we find here, the wavelengths that we use, are wavelengths of non-ionizing radiation, ultraviolet light, that DNA absorbs. Okay, so look at our UV spectrum. It says 200 nanometers to 400 nanometers. I think it goes a little bit farther, but let's just say let's go with that. But I can tell you without hesitation that the maximum wavelength at which DNA will absorb is right here at 260, 280 nanometers. So right about there, 250, right about in there, that's where DNA absorbs maximum. In fact, it absorbs so well there that when we do DNA gels in the laboratory, we can determine bands of DNA by taking an ultraviolet light and shining it on it. We can see that it fluoresces. Okay, so let's talk a little bit about this ionizing radiation. Ionizing radiation works in 2 different ways. We talk about target theory. target is the ionizing radiation acting upon DNA, RNA, proteins, carbohydrates, lipids. It is so energetic, it will break chromosome strands. It's like turning a bull loose in a china shop. The bull goes run through these, just burst everything up. And that's what ionizing radiation will do to DNA. It does this to proteins, it breaks proteins. It will have an effect upon carbohydrates and lipids, but those can be fixed if the DNA is all right, the proteins are right. So it's a direct target, directly affects the DNA, proteins, principles. Indirect has to do with the water that we find associated with living systems. Because that very energetic wavelength will also create some of the very things that we saw with oxygen metabolism. Hydroxyl free radicals, peroxide, and superoxide. These are very reactive, so they start doing even more damage than what's already damaged. Now, there are some organisms that have repair mechanisms that will fix that. They're not nearly as prevalent as are the repair mechanisms that will fix our non-ionizing radiation. I just wanted to point that out to you. All right, well, what are some examples of ionizing radiation? Well, gamma rays, gamma radiation, X-rays, high energy electrons that have been spitted out, beta particles, these are high energy, these are forms of ionizing radiation. We use it in the laboratory, we use it in a clinical sciences for plasticware that has to be sterile, but would not survive heating, we could not subject it to gases. Typically, we would use one of two sources. A cobalt-60 source or a cesium-137. The point sources for gamma radiation. The petri dishes that you use in the laboratory, they're plastic. There's no way they would tolerate an autoclave temperature, let alone higher up, 160 degrees. They have to be sterile, however. So what we do is we simply package them in that plastic and expose them to radiation. Typically cobalt-60. Oftentimes the box that the petri dish is coming will say guaranteed sterile unless they're rodent. Guaranteed sterile unless they're rodent. Because that gamma radiation will pass right through plastic and pass through many different types of metal. In fact, you oftentimes, everyone probably has a chest x-ray or a dental x-ray. Does a dental technician stand right there next to you when they're doing this? No. They walk away. They're typically there's a lead shield someplace. I'll even put a lead apron over you so that you don't receive any undue electromagnetic radiation from that x-ray. They're very penetrating. They're very energetic. They're very penetrating. They're quite lethal. On the other side of the equation, we have non-ionizing radiation, not as energetic. This type of radiation does not even pass through a clear plastic. It is very weak in terms of its ability to penetrate materials. We talk about ultraviolet light, UV light. Now, there are a number of different circumstances that UV light used to be used in, and I can't recall any. I can recall some that I know of right now, but I don't know if you're familiar with them. We're going to talk about laminar flow hoods and biological safety cabinets shortly. They oftentimes will have a UV light in there, so when the operator isn't working in the hood, you can turn the UV light on to sterilize the surface. It used to be in bowling alleys. When you rented a pair of bowling shoes, they placed those into a